Welcome, welcome everyone. I want to thank you for joining us. The webinar today is entitled Targeting the Root Cause of Stress, and our guest speaker is none other than Dr. Thomas Guilliams. So my name is Dr. Lenore Powell. I'm the medical education specialist with Genova Diagnostics, and I'm going to serve as the moderator for today's webinar. We'd like to welcome Dr. Thomas Guilliams. He has his Ph.D., earned his doctorate in molecular immunology from the Medical College of Wisconsin in Milwaukee. For the past 2 decades, he has spent time investigating the mechanisms and actions of lifestyle and nutrient-based therapies, and is an expert in the therapeutic uses of dietary supplements. Dr. Tom serves as an adjunct assistant professor at the University of Wisconsin School of Pharmacy and is the VP of Science for Orthomolecular Products. Since 2014, he has been writing a series of teaching manuals, roadmaps that outline and evaluate the evidence for the principles and protocols that are functional to the functional and integrative medical community. He is the founder and director of the Point Institute, an independent research and publishing organization that facilitates the distribution of his many publications. A frequent guest speaker, Dr. Guilliams provides training to a variety of healthcare disciplines in the use of lifestyle and natural medicines. He lives in the woods outside of Stevens Point, Wisconsin, with his wife and children. So before we begin, if you have any questions or technical issues, please type them into either the chat or question boxes, making sure to send them to the organizer at any time during the webinar. We will try to assist with any technical issues as they arise, and we'll be compiling your clinical questions to answer in the final 15 minutes. The presentation, along with the other materials, is currently available to download. You can access it under the Handouts tab. Simply click the link and the download process will begin. In addition, you have access to the presentation and the slide deck on our website within a few days after the webinar. You can access these resources, previous webinar recordings, brief video modules, and other materials by clicking the Clinicians tab on the home page. Just as a reminder, we have many non-invasive urine, saliva, blood spot, and stool collection packets that can be shipped directly to your patients for specimen collection at home, and the requisition can be completed online. For more information, you can visit your MyGDX account. In addition, online scheduling to speak with a medical education specialist is now available. To access, please just log into your MyGDX account. And now I'm going to turn over the role of presenter to Dr. Guilliams, who again, we would like to thank for joining us.
Thank you very much for having me, Lenore, and the whole Genova team. I've been teaching on this topic for a long time, as many of you know, and I've been recently kind of thinking about it in some different ways. So I'm going to try to focus on something that I haven't really focused on that much, or something I actually have been focusing on in more recently, not so much on the larger question of the HPA axis and stress. So hopefully I can not get caught into the weeds, because there's some technical things that I want to talk about, but hopefully I want to reframe a little bit of your view of the stress response, more particularly reframe your idea of the HPA axis in a more broad way, and then focus on one particular aspect of metabolism that I think is we're looking at more recently, and that is this idea of metabolic signaling and circadian metabolism. So I'm going to focus a lot of my time today talking about the interface between the HPA axis, not so much as a stress responder, although we'll see why we think of the HPA axis as a stress responder, but really more as a metabolic and circadian regulator, and why these things are so sort of critical. So most of you know that I've been kind of for over a decade now, been kind of challenging the notion of adrenal fatigue as really not being the reason or the focus of the stress response. Adrenal function, I mean, when I say the function of the adrenal glands themselves, are rarely a defining feature or root cause of sort of HPA axis dysfunction. So this whole idea of adrenal fatigue is, as we've talked about in the past, it's highly oversimplified, and really it takes us away from those therapies that are really directed at likely what the root causes are, which don't necessarily support the adrenal gland specifically, but really support the whole HPA axis function. And so I'm not going to belabor that point. The other thing I'm not going to talk that much about cortisol. We will mention cortisol throughout, but I think we also overemphasize cortisol as sort of the only part of the stress response. Obviously, it's important for measuring and getting a gauge on what's happening with the HPA axis, but not so much, the end all and be all of the whole thing. And we're going to actually get into that a little bit. So when I ask this question, just more generically, what influences the HPA axis response to, and I put stress in quotes here, and this is a highly simplified version. So I'm trying to give you new categories to think about when you're dealing with patients. So I would say this big category is a response to homeostatic metabolic need. So when you think of the HPA axis, it is an energy need organizer. Hypoglycemia in particular is a very strong HPA axis stimulator. Inflammation is a metabolic dysregulator. It's telling the body something is wrong. Any kind of metabolic dysregulation, and especially we're going to focus today on circadian disruption. These are homeostatic metabolic needs. In a sense, this is the responder part of the stress response. Then we have the anticipation of metabolic need, and this is kind of a unique category that I think the modern human ends up tapping into quite a bit. The psychosocial anticipation of a dysregulation, or a circadian anticipation of a metabolic function. So the circadian anticipation is one that is regulated by the body, but the psychosocial anticipation of dysregulation is not. And that's why sometimes the psychosocial anticipation of dysregulation, meaning I think something bad is going to happen today and I'm stressed out about it, sometimes can overwhelm the system quite regularly. And then the one we're not going to talk about, but I think it's always important to bring up, and it's actually important for chronicity of stress, is adaptations over time from all prior HPA axis activation. So early life stress, this change in cortisol sensitivity that's not related to circadian rhythm, feedback loop inhibition, epigenetics, changes in chaperone molecules, which I'll mention just briefly today, neurosteroid, learned responses, all of these things that happen that cause our HPA axis to begin adapting to these things also influences us. And of course, the longer we have chronic stress, the more this aspect affects us. So the first two are really critical to understanding the stress response, but the third one really tells us how the HPA axis actually is going to function under that. So this diagram comes actually out of my second edition of the book, and kind of shows these four areas of metabolic stress, metabolic disruption, coming from these four categories on the left. The right-hand side says, or the upper right-hand corner says circadian disruption. Then we have in the lower right, inflammatory signaling, the lower left, glycemic dysregulation, and in the blue up on the upper left, we have this whole area of life events. This is that effect, and you can see here, those are influenced by and interpreted through a system of glucocorticoid sensitivity. I have neurosteroids, neurotransmitters, and endocannabinoids. So we're not going to, again, get into the details there, but if you look at the image, the inset image off in the upper right-hand corner, you can see in blue, those three sections of this are the direct homeostatic signals. These are responsive. These are responding to signals that come to the brain, whereas the upper left-hand quadrant is anticipatory. It's being filtered through this psychological signaling that tells the brain what it thinks is going to happen. And that's very important to understand how they're different. Some of you may have heard of this idea of allostatic load, something that Bruce McEwen and his group coined many years ago. And the idea of allostasis or allostatic load is just another way to talk about this tipping point between adaptation and the ability to deal with the metabolic stressors. One of the best definitions that I can give you, and it's something that I think you should keep in mind when it comes to the overall idea of dealing with chronic stress in your patients, is allostatic load is the metabolic cost of reestablishing physiological integrity after a stressor. What does the body have to do to get back to the state where it can deal with that stressor again in the same way that it did the first time? And obviously over time, the adaptation changes. And so that's why allostatic load is often related to the adaptation to stressors over time. And we're going to see here that these stressors may not just be major life signals, they may be the whole aspect of what's going on pathophysiologically with a person, especially if you're dealing with inflammation, circadian disruption, and glucose dysregulation. So again, we're not going to spend so much time on the traditional way, but we're going to mention it a few times here. So most of you know the HPA axis is, basically the hypothalamus converting information and converting that into a signal, CRH or CRF, which goes to the anterior pituitary, which then produce ACTH. ACTH then migrates to the adrenal cortex, where we produce cortisol, and we get this traditional feedback inhibition. So this is sort of, in a nutshell, the very simplified version of the CRH, ACTH, cortisol loop, and that's how most of us have always learned it. And so these inhibitions, you can see the dashed lines that talk about the feedback loops, and we know that if you change the feedback loops, for instance, in the case of patients with major depression, many of them do not sense the cortisol. There's a downregulation of the feedback inhibitors, which then upregulates CRH and ACTH. And that's why oftentimes people with major depression have higher cortisol levels, and their brain doesn't seem to be able to be compensating for that. On the other hand, you see the feedback inhibition being a very strong feedback inhibition in adult subjects with PTSD. And that's why you often see low levels of cortisol in those individuals and low levels of ACTH, because they are perceiving whatever cortisol they're making as much higher. And so these are sort of traditional conceptual ways of thinking of the HPA axis, and that's why we often just measure cortisol and we say we can sort of figure out what's happening from there. And there's nothing wrong with that. I think that understanding how much cortisol is being made is very important in understanding the HPA axis, but it doesn't really tell us everything we need to know when it comes to how this is functioning. So I want you to think differently about the goal of the stress response. Forget for a moment that we're thinking about running from tigers or things like that. Think of the goal of the stress response as, first of all, maintaining effective blood supply of oxygen, nutrition to the brain, heart, cell, muscle. So if we think of that immediate survival, don't think about what's happening. It's an immediate change in nutrient supply, energy supply. It increases energy production. Starts recruiting glucose, free fatty acids, amino acids, so that we can produce more glucose. Glucose is going to be something we're going to need in the short term. Optimizing ATP production for short-term needs. This is all the way that we start thinking about the stress response. But if you start looking at what I'm saying here, all of these things are related to energy, moving energy around, and achieving physiological resilience at the expense of the long-term metabolic reserve. And so, like we think of Bruce Ames's triage theory when it comes to vitamins, think of this as a survival strategy for the cells. And one of the image I give you is these pictures, and actually these pictures come from Germany on the Autobahn, where they're very efficient at moving out of the way for emergency vehicles. The HPA axis is really running on all of these roads all the time, and then has to move everything aside sometimes to get at a stressor or at a metabolic function that's designed for immediate need rather than the long-term survival of whatever those same metabolic functions are using. So thinking about it this way, that the same roads or the same metabolic functions that the HPA axis needs are the same ones that we use for everything else. So when you start thinking differently beyond stress, you start saying, okay, what upregulates or why do we need to upregulate the metabolic system? We need to do that to coincide with diurnal activities of energy needs. So we don't need the same activities all day long. Outside of our stress life, we need certain efficiencies. We need to modulate cell metabolism in anticipation of energy availability. We need to prepare the immune functions for increased vulnerability. And as you know, those of you who study this, you know that the HPA axis is very tied in both in activating and being influenced by the immune system. And then in an emergency, we need to set all these things aside and deal with those things immediately. So when we start looking at this, we notice that when we start looking at the various stress response system, those on the left that basically produce the catecholamines, epinephrine and norepinephrine, which obviously occurs very quickly within seconds. And then the HPA axis on the right, where we see our CRH, ACTH, but we also see the production within this. The adrenal gland is also is also producing mineralocorticoids for, blood volume and blood pressure and other things, as well as DHEA. And some of those are partly regulated through the HPA axis. And so there's lots of other things that are going on rather than just cortisol or glucocorticoids. So we're going to kind of broaden that out, and we're sort of going to back up to the hypothalamus and pituitary more so than the adrenal gland today. So if we start thinking about the hypothalamus, hopefully most of you have studied this a little bit, you realize that the hypothalamus is basically a series or a number of bundles of nerves that are essentially designed to take in information mostly from the body, but also from the external environment, and bring those together into signals that the body can then interpret for how it expects life to be. So this idea of homeostasis, or the idea of regulating oneself, what is the posture? What does the brain think it's going to need? Does it need change in blood volume? Does it need to change its body temperature? What is my heart rate? What is my blood pressure? What are the threats around me? And so that information is coming in and being consolidated into these, you can see here in green, the paraventricular nucleus, or the PVN, and that is basically consolidating most of the information. And one of the other ones I show here in a box is the suprachiasmatic nucleus. The suprachiasmatic nucleus is that part that's getting information from the optic nerve, telling your brain what time it is, or is it light or dark. And those two together create the primary signal that comes out of the hypothalamus to tell the pituitary what to do. Now you can see here on the left is basically our picture of the feedback loops and whatnot. On the right hand side, you can see here is a cell from the anterior pituitary, which gets signals from the brain, from the hypothalamus. And you'll notice that we have a CRH receptor. I don't know if you can see my cursor here, but you have a CRH receptor up in the right hand corner, which then triggers the signaling of the release of ACTH. It also signals the production of the precursor of ACTH, which is POMC. And that's going to be something that we're going to talk about here in a few minutes. But just remember, ACTH is not the only product here, but it is released very quickly. And of course, that's what goes to the adrenal gland and then kicks on CRH. We're not going to talk about vasopressin, which is another signal that comes out of the hypothalamus, which also triggers some of the release of ACTH. We think of that'll take us off on a different tangent today, but I want to focus mostly on CRH and the receptor of CRH, you can see there, and then POMC, which is the precursor to ACTH. So hopefully some of you know this, but a lot of people don't know that CRH or CRF, corticotropin releasing factor versus corticotropin releasing hormone, is part of a family of urocortins. Urocortins are, like I say, a family of compounds that bind to one of two receptors, the CRF or the CRH receptor 1 or CRF receptor 2. And you can see there's some crosstalk between these. So urocortin 1, 2, and 3 are also produced in the brain. And you can see that they will bind to the CRF receptor type 1, as does CRH bind to both of its receptors. So there's a lot of crosstalk between these. Now you say, well, why does that matter to me? Well, as it turns out, CRH is a common, and urocortins in general are a common signal for many tissues that regulate metabolism. And as it turns out, other factors like immune function and whatnot. So we have, I'm just showing you here on the right-hand side, you can see, I'm really mostly focusing in the middle here, where you can see that CRH receptors 1, that's the first column, and receptors 2 in red on the second column. You can see in the central distribution in the brain, there are CRH 1 and 2 receptors all over the brain, including the anterior pituitary, which is what we think of the primary, but you can see it's all over the hippocampus, the amygdala, the neocortex, the cerebellum. all over the place. And in the peripheral, adrenal glands, the skin, ovaries and testes, vasculature, heart and skin muscles, the GI tract. So why are there CRH receptors all over the body and all over the brain? I thought this was a very, straightforward signal from the hypothalamus, pituitary, and then adrenal gland. As it turns out, these signals begin going in many different directions. I don't have time to talk about it today. It's something very interesting to me, and in fact, is that the skin, you'll notice that obviously their skin has some CRH receptors. The skin actually can produce its own CRH. The skin produces its own ACTH, and the skin can produce its own cortisol. So there's almost like a stress response system that uses the same signaling mechanisms that we think of the HPA axis that is completely housed within the skin itself. But for today, I want to talk about metabolism. And as it turns out, we know that the CRH receptors, the CRF you can see here, urocortins and CRH or CRF bind to several of these receptors in skeletal muscle, in the pancreas, likely in the liver and other tissues, and they help regulate glucose uptake, glucose regulation, and much of the metabolic processes. So as it turns out, we don't really know all of what's happening there, but we know that they bind, we know that they influence glucose regulation in the body. So when these are going out, they're not just going to the pituitary, they're going to the rest of the body, creating a different posture. Let's call it a metabolic posture, which is coming out of the stress response system, or at least the HPA axis, because not all of this may be related to stress. It may be related to normal metabolic functions. As it turns out, we know that the immune system most immune cells have CRH receptors. And in fact, mast cells and macrophage are able to produce CRH. So this is a common language or a common signaling molecule and receptors in the immune system. So again, not only do we have a metabolic posture that might be coming out of the HPA axis from CRH, but we have an immune and maybe an inflammatory posture. Shouldn't surprise us if the HPA axis is regulating metabolism. Now let's go back briefly and talk about ACTH. Now ACTH, if you can see in the middle, upper in the middle, if you can see my cursor, ACTH is a small part of a molecule called pro-opiomelanocortin. Again, you can see the name cortin there. It's in this family. And so POMC is what we typically refer to it as. And POMC is this very long peptide, which is cleaved within the pituitary, and then later, some of these are later further cleaved in other tissues. ACTH is just one of many. And so ACTH is able to bind to, you can see at the bottom here, there are at least 5 different melanocortin receptors. The melanocortin receptor 2 is the one that we find on the adrenal gland, the adrenal cortex, which ACTH binds and creates, stimulates steroidogenesis, stimulates cortisol. But look at what you see here. Not only do you see that, but you see many of these other portions of POMC, which then bind to other melanocortin receptors. Those ones in the shaded box in the middle at the bottom, you can see energy homeostasis, energy partitioning as part of the role. So part of the role of ACTH production is the production of POMC, which breaks up into all of these other peptides, hormones essentially, or signaling molecules, which then regulate melanogenesis, sebum production. That's why we have so much relationship with the skin, but also energy, homeostasis, changing metabolism. So the two big components outside of cortisol, CRH and ACTH, are part of these families of compounds which regulate metabolism. So we need to start rethinking about our way of thinking. Could the HPA axis be sort of hijacked by the stress response, but really its job is energy metabolism? And that gets us to this last part of circadian rhythm. So we're going to focus a little bit about this. I'm not going to hopefully get into too much of the details, but hopefully I'll whet your appetite with some papers and some figures that will want you to get more into this. So the HPA axis is essentially one of the main circadian metabolism connections. So most of us know that the light dart cycle comes in to the brain through the optic nerve and through the suprachiasmatic nucleus, which drives sort of the central clock, which then regulates all the clocks within all the different tissues in the body. We're going to talk a little bit about how food does that as well. So again, if you're not as familiar with this, we're not going to get into this too much, but there is a molecular clock that works in the brain, and there's a molecular clock that works, or a peripheral clock that works in all the tissues. And it basically works by, you can see in the box on the right here, a feedback loop essentially that works on transcription and translation of proteins that act as nuclear regulators, and they essentially function on a basically 24-hour cycle. And we don't have time to get into all that detail today, but nonetheless, they work at the tissue level, and then they get resynchronized by signals that come from the brain so that all the body can be synchronized on the same metabolic clock. And so the three main signals that come into the brain are light, and probably I would say that's the biggest one, but here they're shown together. Light, feeding schedule, or the availability of nutrients on the timing, and then physical activity. The other one that's not mentioned in this particular review, which I've added here, is gut microbiota. I believe that there's a signal likely coordinated with feeding schedule, but there's a signal that the gut microbiota sends either to the brain directly or indirectly to regulate the biological clocks in many cells. So I just want to focus on two things that are connecting the suprachiasmatic nucleus and the peripheral clocks, and that is the signals of glucocorticoids, so here's cortisol, and the autonomic innervation. So there's a direct innervation directly from the suprachiasmatic nucleus in the hypothalamus with the adrenal gland, and in maybe other tissues as well. And then, of course, the normal signal of the HPA axis, which is the release of cortisol. And of course, most of you know that cortisol is diurnal. So we'll get to that here in a minute and why that's so important. But okay, so here's a picture of that again. The cortisol, that's our normal HPA axis picture in the middle here. And you can notice in this dashed line, I've showed here that the suprachiasmatic nucleus modulates circadian sensitivity to ACTH. One of the reasons why we have such a profound cortisol awakening response in the morning is because you have two things happening. You have the constant increase of ACTH coming out of the brain, but when you don't see cortisol is only slightly rising right before you wake up, but as soon as you open your eyes and the suprachiasmatic nucleus gets a signal that you're awake, it basically takes the brakes off and allows for an immediate burst of cortisol. And that's why you have that cortisol awakening response. So these two things come together and create these the idea of the circadian rhythm of cortisol. And so and it's obviously not so clean and smooth as what you measure in saliva, because you're only measuring it, let's say 4 or 5 times during the day. Whereas if you're doing something like this, where you're measuring these pulsatility, you have these pulses of ACTH and creating sort of a pulse of cortisol in the body. You might see it as a very smooth curve like you see here, with that jump in the morning of the cortisol awakening response, and that sort of flattening later on during the day. Now we're going to come back to this because these pulses help the body differentiate between a circadian signal and a stress signal, and that's going to be important. And we're going to understand why we don't mess everything up when we have a stressor. Why doesn't our circadian rhythm go completely bonkers every single time we have a stress response? Well, it's good that it doesn't. So this has been known for a long time, this crosstalk between these two. George Crusos and many others have defined this sort of link between the central clock of the suprachiasmatic nucleus and the triggering of the HPA axis on the right-hand side, and this crosstalk back and forth between the peripheral clocks and the central clocks. And that back and forth relationship mostly is defined by glucocorticoids, cortisol, but we're now learning that it's related to other aspects as well. Now, why is that important? Well, cortisol is a major cellular transcription hormone, it turns on and off a whole lot of genes and a whole lot of cells. So managing cortisol becomes, as a signal to the body, becomes very important. Now, today we're not going to talk about the fact that there are at least 6 or 7 ways that the cell can modulate its cortisol signaling capacity, or let's say I should say the cortisol signaling vulnerability. We're not going to talk about like cortisol binding globulin and all the different things that we have there. I think I've covered that in previous seminars, or certainly in my book. So we're not going to cover that, but it's important to understand that if you're circulating cortisol and it has such profound metabolic effects on almost every cell type, it's really important to regulate that in a very important fashion. So cortisol being produced in the morning at high levels and then flattening out during the day becomes a circadian rhythm. And this is actually a figure that I put together, and I apologize because I'm not going to have time to really talk about all of the different components of this particular slide, but this essentially shows a little bit about how the clock genes work within the peripheral clock. It basically binds, like I said, it's basically a transcriptional loop that allows for the production of these proteins that come back and bind to the DNA and then create another 24-hour cycle. As it turns out, glucocorticoid receptors, when they bind to cortisol, they can bind and influence all of this process. So for instance, glucocorticoid receptors and cortisol modulate the peripheral clock by increasing the phase shift of 2 of these called period 1 and period 2. So those are the names of the proteins. And they also reduce the expression of ROR alpha and Rever B. Those are another set of proteins or genes, let's say, that also modulate the stress response. So cortisol affects those. So you have the effects of cortisol on those, but you also have clock genes mediating the acetylation of glucocorticoid receptors. Well, what is the acetylation of glucocorticoid receptors? It changes their sensitivity. So you have a change in cortisol sensitivity or glucocorticoid receptor sensitivity, which is managed by the clock genes themselves. And also 2 of these, cryptochrome 1 and 2, which we call CRY1 and 2, bind to the glucocorticoid receptor. So it's a crosstalk. The clock genes modulate cortisol sensitivity. Cortisol changes clock regulation. And just to kind of give you an idea of how important this is a study that was done many years ago. The details of it are not that important, except to say that this was a study where they basically looked at clock genes in mouse livers, and they basically took the adrenal gland out of some of the mice, and they basically found that over 100 genes that were regulated in a normal mouse all of a sudden became dysregulated, I should say regulated circadianly in sham mice. When they took out the adrenal gland, those 100 genes stopped being managed on a circadian rhythm. 100 genes, and many of these were related to the metabolism, like glucokinase and glucose-6-phosphatase and HMG-CoA reductase. So these are really important energy management genes, which all of a sudden stop being regulated on a circadian rhythm. And so the clock genes within the adrenal gland, is able to influence that. Likely most of this is due to cortisol, but nonetheless, this is a major. So if you take cortisol out of the equation, you radically alter circadian metabolism. And interestingly, like I mentioned, glucocorticoid receptor acetylation is very high in the morning, and that actually reduces cortisol sensitivity. So even though you measure cortisol as being elevated in the morning, as you typically do when you're looking at these things, you can't always assume what is happening physiologically at that moment. Meaning, when you have high levels of cortisol in the morning, you actually have a low target tissue glucocorticoid sensitivity. So the body is able to manage its cortisol sensitivity independent of just the level of cortisol that's out there. And that becomes very important because the body wants to regulate how it's going to respond. You can't just force the system. Now, this is on a short-term basis. When you have chronic elevations of cortisol, this then becomes more dysfunctional, which also occurs when you're giving someone steroid therapy. And we know that you can downregulate the whole process of glucocorticoid sensitivity in the brain and in tissues when you have long-term glucocorticoid sensitivity. For those of you who have been following my teaching on this, you know that one of those major aspects of glucocorticoid sensitivity and alterations in the body is the production of heat shock proteins and chaperones. These are other proteins that bind to the glucocorticoid receptor, and they affect, essentially, they affect how cortisol can act as a transcription factor through the whole process of through the glucocorticoid receptor itself. And as it turns out, these heat shock proteins and other chaperones are also managed on a circadian rhythm. So you have a whole system of circadian biology that's designed to create a very nuanced approach to circadian metabolism. And remember, that's functioning in the normal biology, the normal metabolic functions, as well as stress biology. So then the question becomes, why would I want to use the stress response to help manage my circadian rhythm? Isn't that going to really affect, if I have a large number of stressors, can't I just throw myself way off? And I think that's really important to understand two things. First, the suprachiasmatic nucleus and the pineal gland, which are the two components that get information from light and dark, don't have glucocorticoid receptors. So the body has basically said, hey, we don't want the stress of glucocorticoid, cortisol itself, to outcompete light and the world's clock. And so that's a very important protective effect. So it prevents stress from really getting everything out of whack. However, remember I said that there's a difference between the circadian basal cortisol secretion, basically ACTH, which is pulsatile, and that is different than the ACTH, which is generated from a stress-induced cortisol secretion. And believe it or not, cells can understand the difference between the pulsatile effects of circadian connections and the less circadian or the less pulsatile signals that come from a stress response. And that becomes very important. Now, the longer you have chronic stress, however, the more adaptation you have to the stress, which then influences the circadian timing. So this works really well. The tissues are generally able to tell the difference between a circadian signal and a stress signal. But when you have chronic stress signals that come repeatedly, the ability to distinguish between these two goes down and down. And so interestingly, if you look at a paper like this, which is basically asking the question, how much glucocorticoid circadian clock signals are involved in Cushing disease? So how much of this effect of glucocorticoid overproduction that you get with Cushing disease drives a change in circadian biology, which is likely one of the major factors why Cushing's disease individuals have so many metabolic dysfunctions, because the circadian gene regulation is becoming metabolically inefficient, and that's why you get all the deposition of fat and all the other things that likely come with poor circadian metabolism in those individuals. So then this goes back to ask our question, what is going on? Why does the body need to have such good biological rhythms? And this idea of allostatic load, remember, we're trying to reduce our metabolic cost. So if you properly time your metabolic activity, you can reduce those metabolic costs. So if I modify my heart rate, my blood pressure, my glucose availability, and alertness when I'm going to be active in the morning, let's say, or during the day, I want to make sure I have everything available. So I'm going to have a metabolic system designed for that. I'm going to try to coordinate the efficiency of nutrient transport and metabolism with nutrient availability. So if I'm going to be eating when it's daylight or in the morning, I want to make sure I have all of these factors ready to deal with the activity, the metabolic activity, including the production of reactive oxygen species. So I want to make my antioxidants available. I want to upregulate my detoxification because I'm going to be eating food. Food, other than in modern history, always had potential things that are going to cause me harm. I want to upregulate my immune system so that I can do that as well. So I want to modify my immune system with my feeding activity as well. And then even beyond the daily cycle, I want to coordinate my mating activity with safe birthing seasons. All these metabolic efficiencies change with the food source availability and even migratory changes. So maintaining a metabolic relationship with your environment becomes very, very important. But humans have pretty much been able to divorce themselves from all of these things, including the light dark cycle, but also from the feeding cycle. We don't have to stop eating. Normally, we'll talk here, this idea of a feeding and a fasting cycle becomes a really important circadian signal, but we know that humans don't have to stop eating, and they don't have to go to sleep when it's dark. They can stay up, they can do different things, they can change their clock, they can go across time zones, they can do all these kind of things. So there's and as it turns out, there's a vast metabolic consequence. So here's one of those slides that I don't expect you to get all of, but what I'm trying to do, this is a great paper that talks about the relationship between the metabolic and circadian clocks that are highly differential based on what we call a feed or a fed or an active phase and a fasting or a resting phase. There seems to be a fluctuation in the clock genes and in the metabolic genes that are meant to be turned on and off very specifically when it comes to having available nutrients versus having no available nutrients. And both cycles are needed to synchronize the metabolic clocks in the periphery. So the kinds of signals that we give them is important. And there's been studies, mostly in animals, that suggest that if you look at, some of the signals that come in the Mediterranean diet, like, certain polyphenols and vitamins and phytosterols and certain fatty acids, that those actually are able to turn on and off this peripheral clock in a much more efficient manner than is those diets that we eat in the Western diet. So a high fat, high cholesterol, high stearic acid, high sugar diet. The signals don't seem to be as profound on the turning on and the turning off when we get those signals. So these differential diets, it's not just a matter of when you're eating, as if your brain knows that you're putting food in your mouth, but it's actually the signals that are found in those foods that actually do the work. Now, we could get into the weeds here, and we're not going to do that, but those of you who understand a little bit about ATP production and the formation of NAD, for instance, those are some of the key components at the molecular level. We're looking at how ATP, how energy is produced in the body. And so those are actually some of the signals, the phosphorylation signals that are actually telling the clock genes to turn on and off. And you'll notice that sirtuins, which are a certain class of NAD-dependent histone deacetylases, HDACs, as you can see them here, they require NAD to modulate the body, and they modulate essentially genomic function, and they not only modulate a lot of metabolic function, but they also, as you can see here, affect the circadian rhythm. Now, maybe some of you heard that there are botanicals that are modulators of sirtuin modulators. As it turns out, this was an interesting study looking at Resveratrol, which is known to be a modulator of SIRT1 activity, to see if could they take a animal, in this case mice, that were fed a high-fat diet, could they modulate their clock that they messed up with a high-fat diet? Because basically what they showed in this study was they were able to desynchronize the peripheral clocks by feeding these animals a high-fat diet, and they gave them Resveratrol, and they were able to, let's say, bring the circadian rhythm pretty close to back to what it was even in the presence of a high-fat diet using Resveratrol. Now, obviously, they haven't done this again in humans. So all of the studies that they're beginning to look at, all of these molecules, these phytonutrients that they're starting to look at, they're wondering, can some of these nutrients that we're eating become clock regulators? And they went down to look at the list. You can see here caffeine. Obviously, most of us know caffeine is a regulator, a clock regulator. It can be a clock regulator. But you can see down the left-hand side, cinnamonic acid, DHA and EPA, resveratrol. And so looking at not only dietary patterns, which you can see at the bottom, mostly which are negative, but some of these other ones that may be able to benefit, looking at these kind of things, could some of these metabolic regulators be doing this by re-establishing circadian rhythm? Now, for those of you also who've been into this a long time, you know that the mitochondria are really an important factor as well. And it shouldn't surprise you that the circadian clock related inputs and outputs of the mitochondria become very important. So the mitochondria, if they're the the energy portions of the cell, they are going to be modulated by this whole NAD cycle. And not only that, but it changes the shape and size throughout the day of mitochondria when there's energy available versus when there's energy that's not available. The same idea of feeding and fasting, changing the metabolism of the mitochondria themselves. Now, interestingly, when you look at this, just like you see with circadian biology, there is a vast difference between an acute stressor and a chronic stress. An acute stressor turns out to be the mitochondria adapt very well to an acute stressor. They upregulate biogenesis, upregulate their DNA, they produce ATP, and they also produce quenching free radicals so they can control those free radicals that they produce. However, in a chronic stress environment, the mitochondria really go through a cycle where they're not able to produce as much ATP. They're not able to quench the free radicals, so you get a lot of more reactive oxygen species, and you see a lot more cell death. So just like we see with this long-term adaptation, the longer the body is under, or I should say, the longer the body uses the HPA axis to deal with chronic stressors, the emergency vehicles going down the road, the more the cars on the side just say, it's not worth driving so much on this road. Why am I always sitting on the side? Because we're always dealing with an emergency. The efficiency of these things goes down. So that's one of the reasons why I highly, most of you, if you're working with Genova, you're doing, hopefully you're doing salivary cortisol. And the reason why it's so important to do a diurnal rhythm is because, salivary cortisol is one of the easiest ways to see, do I have a circadian rhythm within the HPA axis? And of course, you can also get a cortisol awakening response added to that, which I always encourage, because that's not only combining the robustness of the circadian rhythm, but it's also telling you about the anticipatory stress of that individual. Now, obviously, you always want to remember that you're only measuring one day, so you can't, predict perhaps too much beyond that one day. And that's why we encourage people to do this on, Monday or Tuesday or Wednesday. People are working from home now, so I haven't seen a lot of data coming out on circadian rhythms with people, working from home after having worked, let's say a Monday through Friday job lately, but I'm sure there's going to be some differences there. But you always want to correlate salivary cortisol with other signs and symptoms, try to figure that out. So we've talked about this idea of responding to homeostatic signals. I said I'm not going to talk about the adaptation on the right, but let's talk a little bit about this thing in the middle, this anticipation of metabolic needs. The psychosocial anticipation of dysregulation can be just as dysregulating, let's say, as an actual metabolic need. And so it's very important as you're getting into this, as you can see on the right-hand side, I've kind of put in this idea of novelty, unpredictability, a threat to self and ego, and lack of control. You can see that sort of band on the right-hand side, because life events that trigger the HPA axis, whether their finances or employment or all the things that I show there, this idea of the novelty, unpredictability, threats, and loss of control, that's how you're interpreting those events. For some of you, public speaking can be extremely stressful, for others not. It's the same exact event, but it has a very different interpretation. And so all of that information is being interpreted, but before the hypothalamus gets that information, it goes through the higher orders of the brain, and it's interpreted, whether it's the amygdala or whether it's the hippocampus or the frontal cortex. That signal is going to be translated through the sensitivity of the brain, which it has from previous glucocorticoid signaling, the feedback inhibition, neurosteroids. The brain makes its own DHEA, for instance, or DHEA sulfate or pregnenolone sulfate. Neurotransmitters, which we know have a major effect, but also endocannabinoids. So all of these act as sort of a chemical milieu, let's say, that interprets these events as being a stressor or a non-stressor. And that obviously becomes very important to those individuals. So when I tell people, how do I create a strategy, you'll notice that I break the strategies for supporting HPA axis function into 3 different areas. On the left, I show central nervous system support. And you'll notice that most of those are in maintaining the appropriate hypothalamus response to stressors. So I would say this is still your biggest lever. Glycemic dysregulation, the perception of stress, inflammatory signals, and circadian rhythm. How much of those aspects of those stressors are you able to help a patient understand how they affect them, and how can you begin leveraging them? If you're not starting there, I would say you really shouldn't start anywhere else, because those are going to be the large signals. You can see at the bottom, balancing neurotransmitters and neurosteroids. Obviously, some of that can be done with helping a patient understand their situation, but obviously there's some things you can do by modulating those with diet, physical activity, and supplementation of various sorts. If they don't have enough DHEA to counterbalance the cortisol, you can look at that as well. On the right hand side, something I didn't talk about today, only in part, is how do I manage the target tissue cortisol modulation? How do I modulate, for instance, the hormone 11-beta HSD, which converts cortisol to cortisone, a major factor in recycling activated cortisol in the body? There's things that we can do about reducing inflammation, reducing central adiposity. We can address that. How do we increase heat shock protein modulation? And perhaps certain plant adaptogens are able to do that, but probably circadian rhythm is another part of that as well. And then only in the middle do I have this idea of adrenal support. And I put in question marks whether we have much of this. We don't really know that much about supporting the adrenal gland itself. We probably can do that with a lot of the antioxidants we're using, but I would suggest that most of the benefit that we have is in affecting at the brain or at other tissue level, and only secondarily are we dealing with adrenal support directly. So always remember, when you're looking at this picture, always to be dealing with asking the question, what is the brain perceiving as a stressor? And most likely it's going to be in responding to a metabolic need, anticipating a metabolic need, or in adapting or the long-term adaptation to responding to one of those needs. The second edition of the book, which was published last year, includes a lot of more detail, and I've included a whole chapter on endocannabinoids, and I've expanded the effects of stress on memory and a number of different things, and probably upgraded and tried to include a lot of new information that wasn't there in 2015. So hopefully there's some questions for me or for others, and Lenore can hop on and answer some of those questions. So thank you very much for your attention.
All right, so phenomenal presentation as always. I just have to say my mind is a little bit blown right now. You have my mind just going in many directions because, really kind of balancing out this peripheral and central clock balance here, and just understanding that it is, as you mentioned, not all just about cortisol. If you could go a little bit more into detail, you did mention certain foods like your standard American diet versus your Western diet and how that can throw off the clock. But what about timing of foods? What about those individuals who are doing, for example, one of these diets in which you fast for a long portion of the day, like intermittent fasting? How would that possibly throw off that peripheral central clock regulation?
So I mentioned the idea of feeding and fasting and the timing of nutrient availability. So I kind of, again, with the time I had, I didn't spend too much time on that, but the timing. So your body obviously knows when you have nutrients available and when you're fasting. And I talked about the fact that, a lot of Western subjects are never really truly fasting. We eat so much and we're off circadian rhythm. So when we start thinking of intermittent fasting, and I specifically want to talk about it within a 124-hour cycle. So let's say, the 16/8 or whatever people are doing as far as, when they eat during a day and then when they stop eating during the day. I would say that one of the challenges that we have, which is socially driven, is most people skip breakfast in order to shorten their window. A lot of people like to be at 8 hours or let's say 6 hours or something like that. And so in order to deal with that, and they want to eat dinner or something with their loved ones, they skip and they don't eat the first meal until noon or something like that. There's a lot of debate about whether that causes, weight loss, and that's where most of the research is at. But I would suggest that if you look at the broader research that looks at circadian gene expression, that approach is really the wrong way to do it. In my opinion, the bulk of the data suggests that when we wake up in the morning, our body is anticipating food and it's also anticipating activity. And so the window of eating should occur not long after daylight begins, and probably I would say the eating window should be longer in the summer and shorter in the winter, but also our physical activity should be more in the summer than it is in the winter. So what messes part of this up is that some people are trying to do everything by changing when they eat, but they're not physically active, or they're trying to, alternate when they're physically active when they're eating. So I suggest that biology tells us that we should be eating when it's daylight hours, and we should be physically active when it's daylight hours. And the synchronicity of our clocks are going to be stronger and stronger. As far as a weight loss, because when you look at these studies, it's very hard to manage caloric intake as well as all these restrictions, because some people, even if they have a shorter window of time, don't consume as much calories as they would if they were able to, eat all day long. So there's a lot of confusion when it comes to intermittent fasting data, partly because most of that data is looking at weight loss rather than looking at circadian biology. So I suggest that people should be fasted for at least 8 hours in a 24-hour cycle, and most of that can be while they're sleeping. But if they're gonna narrow the window more and more beyond what they normally would do, I would take the time off the back end of the day rather than the front end of the day.
Amazing. And it also makes you wonder, just based on what you presented just now, if potentially the hours fasting versus the hours eating should actually be different, depending on what season we're in. Just kind of feeding off of that daylight versus a lack of daylight that we see in the summer versus the winter times. You mentioned quite a bit about, well, you mentioned very briefly something about resveratrol and how that, despite having a high-fat standard American diet, can help to bring back into synchronization the peripheral and central clocks, as well as other phytonutrients. And so what I thought was kind of interesting is that when we look at the various nutrients that we usually use to support adrenal health, sometimes we do include those phytonutrients. And I'm wondering if they're working more on the synchronization of the clocks versus directly on cortisol now. What do you think about that?
Well, okay, so that was of course that was an animal study, but so I've done a lot of teaching, and there's been a lot of studies that have looked at taking, so the peripheral clocks are those that are in all the, all the tissues, primarily in, when you're looking at them, you think of the liver, metabolic tissues, or in the skeletal muscles, things like that. And so you can get a dissynchronization between the central clock, which is the light dark cycle, and the peripheral clocks just by shifting the timing of food. So for instance, they've done studies in humans where they keep their light dark, they make them wake up at the exact same time every day. They put them in a lab, but they shift when they eat, and they can show in a very short amount of time that what's happening in the brain is now a dissynchronous with what's happening in, let's say, the liver. And that study in animals was in peripheral clocks, meaning it was the metabolic clocks were being shifted with the high fat diets, and they were able to, in a sense, resynchronize or partially resynchronize the peripheral clocks back to closer to the light dark cycle when you gave them resveratrol. So the question becomes, and likely it's because you're helping to resynchronize the production of NAD, the sirtuins, which influence that whole clock. So the question, I totally agree. I think the idea of phytonutrients, we talk about adaptogens, we talk about sort of these, we don't really know a lot about how these things work. We just have some outcomes. We don't have a lot of biomarkers that we can test for adaptogenic function. And I think you're right. I think a majority of them are likely functioning at the level of the brain, or some feedback inhibition, or in the case of heat shock proteins, which change glucocorticoid sensitivity, or circadian rhythm, like we see here with resveratrol, all of which then does affect cortisol output, but not in the direct way that we think. It's not in measuring it from a stress standpoint. It's like recalibrating all of the receptors and all the systems that then tells us when cortisol should be there. And that's why, again, understanding when you're doing a salivary cortisol test, especially when you're adding the cortisol awakening response component to it, you really want to understand what you're measuring, what the day was like that you measured in that subject, and then the nuances of, circadian rhythm. I tell clinicians, having a low or a high cortisol overall during the day is much less important than knowing whether it had a circadian rhythm. If you have low, let's say the cortisol overall looks good, but it's flat, that's actually worse than having high or low cortisol. If you have somebody with a low cortisol level, but they have somewhat of a good circadian rhythm, meaning that most of it's in the morning and it flattens out, that tells you that you have some responsiveness in the HPA axis that can still be, that's still responding to a circadian rhythm. If you have somebody that's flat, and that is more problematic, because it means that the responsiveness to the whole circadian rhythm is either lost or extremely low.
There are so many more questions that I have to ask, but in the interest and just being very respectful of your time, I think we're just going to end the question and answer session right there. Again, great presentation. One thing, Dr. Guilliams, that I really appreciate about you is that you are not afraid to challenge something that we currently accept and back it up with science and literature, and just kind of make us think about things a little bit differently. And I would say that today you definitely made me think about the HPA axis and the function and how to address it with various patients. You changed how I'm going to be looking at that from now into the future. But Again, we're going to end our question and answer period there. For additional educational materials, you can always go to the website gdx.net. You can contact customer service as well. As many of you know, we do offer complimentary appointments with our medical education specialists to answer questions related to our testing, including choosing the right test and reviewing patient test results, which you can now also book appointments through our online scheduling system. And so we also would like to encourage those who have not registered for our upcoming webinars. You can always do that on the website, as well as if you have not checked out the Journal of Diagnostics podcast called The Lab Report, focused on functional medicine and specialty laboratory testing, natural therapeutics, with hosts Dr. Patty and Michael, and an assortment of guests, I would strongly suggest that you go ahead and check that out. Again, Dr. Guilliams, I really want to thank you for such a great presentation, and I think it's time for me to get the second edition of the book. I have to learn a little bit more.
Well, it's my pleasure to be with you.
All right, you have a wonderful day. Bye-bye.